ये पे लॉगिन है सर Zoom लॉगिन
जॉइन किया सर
ये
इसमें आवाज उसको ऑफ कीजिए
साइन
सर हमने वो भी सही भविष्य को डॉक्टर से
Please focus now. Can you start? Sir, is it audible? Hello. Good afternoon, sir. Is it audible to you? Can you start share your slide so that others can see that, or we want to just check. Sir, please unmute yourself.
Okay. Now you are getting?
Yeah, it's audible now.
Okay.
Yeah.
Okay.
Can you start share your screen?
Share screen. Okay.
We want to. Yeah.
Okay. Then I have to select files, sir.
Yeah, just you have to open the PPT. After opening the PPT, you just start share screen and click on the PPT. It will display.
1 second. From my desktop, I can share it.
Yeah. So actually this Windows is visible. You close the window. After opening PPT only you have to start share the screen.
Okay. Now okay, sir? Is it now okay?
No, actually you have to close the window. No. Stop sharing.
Okay. Okay.
Stop. Exit to the share screen.
Okay.
Yeah. Now you can click on share screen and to the point PPT. You can point your PPT. Now you can once again you start share screen, sir.
Okay.
And click on the opened PPT already.
Yes. Now okay?
Yeah. Yeah, it's visible now. Yeah, that's fine. Yeah, that's fine. Okay,
No?
so I'll formally start.
Now it's
Yeah,
okay,
it's
sir?
visible. Yeah.
Okay.
Yeah, okay. Okay, dear participant, I welcome once again this afternoon session. Good afternoon to everyone, respected afternoon speak. So then, dear participant, good afternoon to everyone. Today afternoon we have Mr. Dhaneshwar Reddy, Managing Director, MICCO Laboratories, Private Limited, Chittoor District, Andhra Pradesh. So it is very nice to have Mr. D. Dhaneshwar Reddy is acting as a technical and managing director at MICCO Laboratories Private Limited, Tirupati, Chittoor District, Andhra Pradesh, India. He has completed MTech Biotechnology in 2008 from JNTH University, Hyderabad. He has 10 years of industrial experience in plant biotechnology and 9 years of teaching and research experiences. He published 8 research papers, 2 books, and presented papers in the conferences. He guided 18 students for MTech in biotechnology and many students for BTech, MSc, and BSc biotechnology. He worked as assistant professor and member of Board of Study in Department of Biotechnology, Sri Vidya Niketan Engineering College of Alpha and Alpha College of Engineering and Technology in Tirupati and Hyderabad, respectively. Scientist and manager in Biologist Hyderabad during 2006 to 2008. Served production manager in AG Biotech Laboratories Limited, Hyderabad during 2000 to 2003. He worked as a guest faculty and course in charge to MSc Microbiology and also served as a subject expert BOS of Biotechnology Department of Sri Ramakrishna Degree College, Nandial, Andhra Pradesh during 2004 to 2006. He's a life member and fellow in many societies and also acting as a reviewer for Journal of Analytical and Bioanalytical Techniques and Journal of Biosensors and Bioelectronics. Dear participant, we have a very rich, experienced person in the field of this tissue culture. I hope his teaching will bring you a lot of scope, how to start, and a lot of challenges and the opportunities there in this banana tissue culture. And this will be the good news for the young entrepreneur who are seeking to start this tissue culture laboratory. So in future, most of the plants like disease resistant and pest resistant and stress resistant plants can be produced through tissue culture. So I request every participant to take part actively. And if you have any question, please leave it in the chat box. We will answer it at the end. So once again, I welcome sir.
Thank
Sir,
you, sir.
this time now you can start your lecture.
Okay, sir. Sir, thank you for giving this opportunity to all persons. I'm here today to discuss about scope, challenges, opportunities in banana tissue culture industrial techniques. Before I start, here I am going to give a brief introduction about my organization, my industry. I started this industry in 2015, June 6th, in Tirupati, Chittoor District, Andhra Pradesh, as a MICCO Laboratories Private Limited. And the company facility, lab facility, tissue culture lab facility was accredited, recognized by the National Certification System for Tissue Culture Raised Plants, NCS-TCP, Department of Biotechnology, Ministry of Science and Technology, Government of India, New Delhi. And this company has strongly focused to produce quality products through a different innovative approach processes and give a good quality services to the farmer community. And the company also have some of the activities who are interested to start a tissue culture laboratory. Will give a support how to design a plant tissue culture laboratory and design a acclimatization facility. It is the hardening facility, and to design a greenhouse facility, to design a shade house facility. It is the low cost technology which is generally using in all commercialized tissue culture industries. Not only this, a few students are, some of the interested persons are looking to training. For those candidates, we are allowing training programs and also a research facility we are also giving. And some of the take up of turnkey projects. And a few people are looking to production of some light plant material through the contract production. This also we are giving. And we are producing elite plant material of only banana we are producing. This about the organization. And before start, scope, challenges, opportunities in banana tissue culture. What topics today I am going to discuss on this. The topic wise, here everyone talking about a banana. Banana, everyone knows about different methods we may follow, vegetative method and other new approach method. It is the tissue culture method. What is banana? When it starts, the origin of the banana, survey of the banana, which varieties are generally growing globally, and in states of different states of India, what are the propagation methods of banana in different places of India and worldwide. Based on these two topics, we'll see the scope of tissue culture banana. Is there any need or any gap, producer and consumer? And what are the facilities are required for developing a plant tissue culture, particularly for production of the banana plants? And what's the technology which is involved to produce disease-free elite plant material through the tissue culture technology? And what are the problems generally facing in tissue culture technology? It's all based on my experience here. I am going to discuss. I am here. And what are the major challenges for major challenges in banana tissue culture, financially, technically, marketing, and politically? What challenges may involve? And finally, the SWOT analysis, like what is the strength of the banana tissue culture? Not only strength, what are the weakness and opportunities and threats? This gives the complete analysis of the SWOT. So based on this, can I start a small scale industrial plant tissue culture lab? So these are the topics, 7 topics here I am going to discuss with you. Let us start about the introduction. The banana started from the Southeast India and move on to Philippines, Malaysia, and globally. This banana is a globally very important crop because of the major product is producing globally about 97.5 million tons of banana producing, This is using for livelihood of million of people, and it occupies 20% of the total area under crop of India. Total food production it occupied, it contributed 37%. And this banana is tropical crop. It required the temperature 18 degrees, 13 degrees to 38 degrees. Relative humidity it required 75 to 85%. Generally, it required a good drainage for development of a banana plant in a field, and humus-rich soil, very fertile soil, and moderate moisture content is also reached. The required pH level in the field 6 to 7.5 pH value. It is very preferred to grow a banana in any field at a tropical level. Suppose at a higher temperature level, all bananas are going to see the scorching. And if you see the high wind, so this damages the entire crop of the field. So this more major difficulties to extending the area of under cultivation of banana is non-availability of disease diagnosed planting material. Means through the conventional process, whichever plant material using for propagation of banana in a field, we need diagnosed plant material. That is not possible. But if we send the material to any laboratory, they can screen only one circuit, not all circuits. This is the major issue facing. And then what are the major countries worldwide producing? The country like India, first place occupied, the producing about 30.47 million tons of banana producing, and it states globally 26.7 is the first country occupied producing from the all states of the country. The other countries like almost all USA, UK, remaining all countries above mentioned, all 32.67 million tons of banana producing by other countries, and it occupied 28.6%. So after India, it occupied by the China, Indonesia, Brazil. Like these countries are producing more banana products. And then next slide shows the state level in India. First place occupied by the Gujarat. It is the first place occupied by the Gujarat, 4.187. Second place going to Andhra Pradesh, 4.14. The third place is Tamil Nadu, 3.64. The last place goes to 1.22. This is banana production in different states of India. It is as per the National Horticulture Board, which presented in 2017. Then area and banana production at different states of in India. This is also published by the National Horticulture Board Mission in 2017. Here area by first place occupied by the Andhra Pradesh, not Andhra Pradesh, Tamil Nadu, Karnataka, Kerala. These three are in first, second, third places. The other states are occupied 178.52. Gujarat is also occupied 64.709. What varieties are generally preferred for propagation of banana? It is a conventionally or PTC level. Generally important cultivar Robusta. This also Tamil Nadu, Andhra Pradesh, Karnataka, then Madhya Pradesh, Maharashtra, in different areas of Uvan, Nendran, red banana. See, the different states, how they are growing different varieties of banana, particularly Andhra Pradesh, Dwar, Cavendish, Robusta, Astali, Amritpani, like this Assam, Bihar, Gujarat, Jharkhand, Karnataka. 2 or 3 species are more commonly using for farming of banana plant species, like states like Kerala, Madhya Pradesh, Maharashtra, Orissa, Tamil Nadu, West Bengal. These states also growing well banana plants, and these are the shoes red banana, Robusta, Rastali, Alayankodan, Nendran, Cavendish, Montan, Safed Welchi. But the first place occupied globally is the Grandland variety. That's why I selected this variety practicing in my tissue culture lab facility. Why this selected tissue culture laboratory? To propagate these banana plants. So nowadays, tissue culture facility occupied, it's a very important role playing in science and agriculture. Due to through the tissue culture propagation technique, we can produce disease-free plant material, high yielding varieties. So that's why this tissue culture technology is commercially very important role playing in science and technology. What propagation methods generally preferred? The propagation methods like vegetative propagation methods. Generally, everybody knows about bananas cannot produce any seeds, but a few technocrats are extracted seeds. Those are growing. Those are using for development of a banana plants. And the main propagation method for vegetative propagation, the plant material used for banana practices is the sucker. Its name is rhizome. Generally, these suckers are again classified into sword suckers, water suckers. Sword suckers these are conical structure and narrow shaped leaves, but these water suckers have a broad leaf material and a narrow shape stem. This cannot be used. These are far away from the mother plant. And generally, the suckers allowed are collected from the mother plants, generally 500 grams to 1000 grams, which are suckers used for practicing in tissue culture laboratory. Generally for propagation of vegetative propagation practices. And then a major constraint for this propagation technique is the readily availability of the large quantities of spot suckers is the very difficult. Suppose, for example, one field, their plantation done 4,000 plants in a 3.5 acre area. Almost 80 or 85% of the banana plants may give high yielding. So the selected mother plants are 1,000. Every selected mother plant has a different characteristic. Means in the way of quality, in the way of yielding, and also non-availability of swot suckers. And these suckers also sometimes affected by some disease-causing agents like bacteria, fungus. When we select and taken and plantation done in our selected field, and also their size is also different, not in uniform. And the other method is tissue culture. Generally, in tissue culture, from the selected mother plant, a piece of material collecting, which is grown in a artificial media under aseptic conditions. This piece of material we may call as explant. To collect this explant from the mother plant, we have to see that the healthy of plantlets, disease-free plantlets, uniform plantlets, high-yielding plantlets, and 2-2 type plantlets. This will be happen through the tissue culture technology only, not with the vegetative propagation method. Of course, in vegetative propagation method, healthy plants are available. Can't say these plants are disease-free because of some soil-borne microorganisms may cause the damages of circuits. Some of the endogenous bacteria colonies may also secreting and damages the plantlets. In this tissue culture technology, major technology is applying is that micro propagation technique. This is the technique where we can propagate number of propagules at a small scale level in a small area. This is the possibility. This is happen only through the tissue culture technology only, not with the conventional practices. And also tissue culture banana planters are made from selected high yielding mother plants, which is the best plant. Not only banana, take any ornamental hardwood species, flowering plants, you have to see the all physiological parameters. Is it suitable to develop new clones or new propagules through the tissue culture technology? Then only we have to select those plants. Then screen the plantlets, screen these mother plants, suppose say for virus induction. Screen any kind of viruses are occupied in these metabolic pathways. You have to study and all select these all mother plants. What are the advantages of this micro propagation technique? Generally, you can produce firstly disease-free plants. We can produce disease-free means fungal-free, bacterial-free, and viral-free plants will can be generated through this technology. And we can produce a larger number of uniform propagules in a small area within a short period of time. You can generate millions of propagules through this plant tissue culture technology in a small area. The growth is also uniform. It increases yields because of if we take any part of the plant material from the mother plant up to the limited cycles, they can generate very uniform rate of plantlets at a genetic level, any level, product level. If we cross the number of cycles, we will see the different challenges in tissue culture technology. We will discuss later. And also we will see the early maturity of a crop. We can reduce 20 days or 25 days. There is a possibility availability of round the year this tissue culture plant we can take at any time throughout the year. Through the conventional propagation process or practices, it is not possible to allow the mother plant's next generation ratoons. But this tissue culture technology will see the very successive ratoons, generally 2 successive ratoons. In banana tissue culture practices, every farmer or every successive customer, they are getting 3 crops, 1 main crop and 2 ratoons within 30 months of duration time. Almost all plants bear bunches, 95 to 98%. This not happen in vegetative propagation method. Not only this, if we want to produce a new variety, when I completed my MSc Biotechnology in 2000, I joined in Biotech Laboratories as a technical assistant. At that time, only producing dwarf Cavendish variety, Robusta banana varieties, Gerberas, number of species, yellow, green, mixed, white, forestry plants like Indian teak, Gamber teak, Paulinia, different teaks. Number of orchids, philodendron, dendrobium, number of flowering plants producing that in that company. So now we'll see the globally which banana variety occupied. No one asking about the dwarf Cavendish varieties. Everyone looking to only granite variety. A few states of India they are asking Robusta. Globally also is more demand for granite variety. A few species are locally demand. That's why few areas are asking particular like red banana, Yalaki, Amrutapani, Robusta. Few people are asking Puven. These are locally market demand banana plants. So this is also possibility. You can generate a new variety which is locally adaptable, locally useful. You can see at a time globally and locally national, international level. You can develop different varieties of plant species. They can introduce and multiplied, and we can go. You can see the contract production also here. So from this introduction part, from this survey of the pot, from this variety of the pot, then we'll see the importance of banana in tissue culture and its scope. Generally, it's a tropical plant and which is very important horticulture crop in India. Totally complete production capacity in our country to produce tissue culturized banana plants. Almost is 650 million plants per annum it's required. But how many companies are involved? There are different tissue culture laboratories are involved in production of banana varieties and ornamental varieties, flowering plants, and different so on species, particularly when we talk about banana plant. We can divide in India tissue culture facilities, tissue culture companies into two groups. One is recognized companies, the other is the non-recognized companies. Recognized companies are DPT recognized companies. National Certification System for Tissue Culture Facility. The DPT, Department of Biotechnology, Ministry of Science and Technology, providing this certificate. After observing complete all criteria based on this, they are providing a certificate. In India, there are only just 109 tissue culture industries are recognized by the Department of Biotechnology, and a number of laboratories are working to produce variety of species, not only banana, all kinds of varieties. Those are non-recognized laboratories. They have the technology. They are following all whatever handling practices are required in tissue culture facility. Everything they established, but they are not recognized. Almost 320 or 319 tissue culture laboratories are existing in our India. But these all companies are producing just 455 millions of banana. See the gap, how much is required and how much is producing. And a few farmers are generally, they are preferring this vegetative propagation method. They are using suckers, and some majority of the almost 90-95% of the farmers, they are looking to banana tissue culture plants. In India, the demand is 3,436 millions of banana plants. So there is a huge potential to produce banana tissue culture plants to meet at present demand, not only in India, globally. If you want to start a tissue culture laboratory, what are the facilities are required? So the components of the plant tissue culture laboratories are classified into two types: non-sterile zone and a sterile zone. This non-sterile zone having different sections: washing area, bottle storage area, media preparation area, sterilization area, grading area, acclimatization area. Sterile zone, it occupies with the different sections like dress cubicle room, media storage room, culturing room, or inoculation room, growth room. Every section is playing a critical role at the industrial level. Generally, washing area, there we are cleaning the bottles. We may call culture vessels. In any commercial laboratory, the culture vessels capacity 300 ml or 200 ml, 250 ml capacity of culture vessels. Those may also called as charts, used for culturing practices. Washing of these bottles, reusable bottles, not using any plastic containers. To maintain cost of production, any commercial laboratory preferring this kind of bottles. So there is a protocol to wash each individual bottle. Suppose if any bottle is contaminated with fungal species or bacterial species or any other debris material, for this kind of bottles, there is a separate protocol. So different protocols are following to wash the bottles at industrial level. Otherwise, we'll see the major issue. Why means here, if any sedimentation material occupied inside the surface of the bottle, these bottles are using for media preparation and going to sterilization process. This sedimentation material is combined with our media components and which is going to changes its characteristics, so it may influences to grow of the any bacterial colonies. After culturing the plantlets onto the surface of the media only. This problem here I observed in my industry. Like this way, contaminated bottle also. There is a separate protocol for contamination bottles to wash them in a washing section. You can't directly use this or collect these contaminated bottles to washing section. Firstly, we have to kill all kind of microorganisms and their spores through the sterilization technique. Means after the sterilization process, only all contaminated bottles are going to the normal washing section protocol. Then after washing up these bottles, then we are keeping the bottles in bottle storage area for next preparation of media. Next to non-sterile zone, it is the very important, this section is playing very vital role in preparation of media. In media preparation room, different types of equipments are essential. Equipments like pH meter to adjust the pH value of a media, generally in tissue culture. The media using MS medium 1970-62 modified media. It contain all types of macronutrients, micronutrients, and vitamins. We are changes doing in only growth hormones. Suppose for initiation, the ratio of growth hormones we are changing. Initiation means the explants which are going to practice through the tissue culture technology or to develop any plantlets. So there changes in cytokinin and auxin ratio, like the way multiplication of any propagules means raising the culture in production department. In this practice, multiplication is the major important step to stock the cultures to meet the demand. So here also cytokinins are playing a very important role. And then shoot elongation purpose, we are preparing different types of media. Here cytokinin and auxin both are playing an important role. Then root development purpose, generally preferring auxins, only auxins. Few persons are adding cytokinins, less amount, less concentration. So different types of media compositions. Here we are formulating, standardizing, and finally we are completely adopting this technology in a particular industry, plant tissue culture industry. And autoclave is very important to eliminate any contaminant. Their spores which are present in RO water or any media chemical component or any surface of the bottle. And balances, homogenizer, a few equipments very important in media preparation. Homogenizer for complete homogenization of a prepared media. This is about media preparation room, where we are going to preparation of the media for specific plant species. And then sterilization zone, they are also sterilizing the entire media. Grading area. Well-suited rooted plantlets are giving to hardening practices. We are sending these plantlets to the grading area there, complete removal of the agar from the plantlets and sending to the acclimatization section. Acclimatization there, acclimatization of the immediate plantlets in the area through the greenhouse facility only. It is the low cost technology where we are allowing tunnel systems we are maintaining. Their humidity and temperature maintaining continuously, allowing for 1 month. After 1 month, then shifting to secondary hardening. So 2 sections are generally very important in acclimatization area. Final section is the dispatch area. So well grown 1 feet plant or 4 or 5 leaf plants we are delivering to the farmers. In sterile zone, dress cubicle room to avoid contaminant from our surface of the body. Generally, we are carrying the different types of contaminants. We have to avoid to enter into the sterile zone. So, dress cubicle room, there we are keeping the all aprons, mask, caps, slippers, everything, and leg wash, hand wash, everything there. Then next important section is here, sterile zone media storage room. There we are keeping the media for 48 hours to observe any contaminants growing on surface of the media. Means here to the washing section, to the media section, any problem is raised, identified before going to the culturing practice in media section. Inoculation room, generally in inoculation room, trimming the culturing, shifting the culturing, subculturing practices done in inoculation room. In inoculation room, generally laminar airflow benches are using. In front of the laminar airflow bench, only all cultures are opening and doing the subculturing practices. Trimming means any dried cultures, waste material, or any roots are developed, separating big plants, big plantlets, medium plantlets, small plantlets, like that. The complete inoculation practices done in inoculation room, then growth room. It's very important room in sterile zone where cultures are growing in this room continuously 24 hours round the year, 365 days. We are maintaining the temperature and humidity under controlled system completely. 24 plus or minus 2 degrees temperature around the time. Humidity generally 55 to 60% humidity will be maintained in the growth room. All cultures are growing inside this growth room. See some of the photos here. Section of the washing area where few tubs are using to clean up, to wash up all the bottles, followed by preparation of media room. There we need different kinds of the instruments which are using for preparation of media. You are seeing here homogenizer. This is the autoclave, and here you see the pH. This here closed two balances are occupied. This one is pinched off centrifuge. Centrifuge. Beside that, there is a other equipment is there. This is the freeze to store the all stock solutions inside this freeze. Freeze is also important. This is the media storage room, which is occupied in sterile zone, where we kept for observation for 48 hours to observe any kind of microbial colonies on surface of the media. This is the inoculation where technicians are going to culturing practices in front of the laminar flow. The capacity of this company, 6 technicians are working at a time. Per annum, we are producing 12 lakhs of banana plants. Per month, we targeted 1 lakh. From next year onwards, we are enhancing this. This is the growth room where cultures are growing in this. I'll see the bottles which are in green. Under the lights, rooted plantlets are growing. Under the dark, multiple cultures are maintained. This is the hardening facility where immediate vast plantlets from the grading area. Plantation done here. Here you are seeing tunnels. These are the tunnels covering with polypropylene sheet when you shifted immediately to the primary hardening zone for 3 days. After 3 days, then remove the PP sheet so that it maintains temperature, humidity. If the temperature is going to rise up, we are going to close this tunnel. So for 20 to 25 days, it's important to maintain the humidity and temperature inside this primary hardening area, which may call a greenhouse. Whatever propagules are developed through the tissue culture technology, these propagules are going to first acclimatization step to the environment. So you have to take care of all these important factors like temperature. If the temperature is high, the plants are going to dehydration. We'll see the more mortality in primary hardening. This is the secondary hardening. In primary hardening, the plantlets are allowed to 25 to 35 days. Now these are called as primary hardened banana plants. Plantlets are turned to now first acclimatized plants. Then these plants are now shifted to secondary hardening. That means here 4 by 6 size of poly bags we are using. These poly bags are filling with soil mixture. The composition of the soil mixture generally we are procuring locally available red soil. FIM, cocoa peat, some other we are adding and preparing the soil mixture filling in these poly bags using for growing of primary hardened plants. In primary hardening, the plantlets are generally here allowing with a cocoa peat material. It is the sterilized cocoa peat is using for growing up this plantlets in primary hardening. Next, what is the banana tissue culture technology? See, the flow chart may explains the entire banana tissue culture technology. It starts from selection of the mother plants and to the dispatch of the tissue culturized plant. Selection of the mother plant, it's a very important role playing to produce healthy, disease-free, 2 type disease plants. Followed by selection of the dotted swot circuits. These are going to the next surface sterilization and going to the inoculation step. And then establishment of cultures in establishment of cultures and origin of new shoots. Multiplication of shoots, elongation of shoots or rooting, primary hardening, rooted plantlets, secondary hardening, rooted plantlets, ready to dispatch. So, globally, number of technologies are available for micropropagation of any kind of species, not only particular banana, or commercialization of any plant. This technology divided into different stages. Previously mentioned that. The first step, selection of the mother plants, mainly depends on the commercially viability of a plant species. We have to select a mother plant. And then second is the feasibility. Technology is adaptable at the laboratory level. Execute the technology at a production level. Produce the product quality level. So this is possibility. This is possibility through the elite healthy disease resistance or any kind of free plants, and also high yielding variety of plantlets. These plantlets, if we selected from the other plant, are going to screen the virus induction. So this is the other plant we selected. Few characteristics of the mother plants, we are collecting a data. Which date we are going to collecting? And date of success collection. Who is the farmer? Name of the farmer and the village name, mandal, district, his contact number, which plant variety growing, and the general information, the type of soil, type of irrigation. Is it a main crop or a return crop? We are checking on all. Plant height. Generally, after selecting what is the height of the entire plant in centimeter, generally ground 9.6 centimeter. Plant get the base level, plant get at the top level. Number of leaves at the shooting level, at the bunch level, the complete bunch level, number of hands per bunch, number of fruits per bunch, and then length of a fruit, first hand, last hand, weight of a bunch harvest, number of swords occurs, number of waters occurs. Is there any pest or is there any insect or any kind of diseases or any other information? Based on this, we are selecting the mother plants. After selection of the mother plants, we are picking the heads growing in shadows under controlled conditions. The collected suckers are now, you see here in shadows like this. Now these mother plants, now we treated as mother plants, going to surface sterilization process. This we may call as explant. Choice of explant, this may depends on variety of species which is going to propagate under controlled conditions. For banana plants, we may collect the rhizomes. For teak plants, internodal parts, leaf material, roots, center bulbs, any type of other organs you can select as a explant. This is the explant which is collected from the mother plant and used for surface sterilization practices. So here we see the sword suckers and water suckers. Then these selected suckers are going to surface sterilization practices. At nursery level, we are collecting the explants. These are going to wash to remove the any debris material from the roots or from the surface occupied the explants, a layer of the plant material. Now these plantlets are treated in Bavistine solution or dithenium 45 solution per liter, 2 grams per liter, and a few drops of twin 20. Submerging these explants in this solution 45 to 60 minutes. And then the next step follows in inoculation room. Their explants are rinsing with sterile distilled water for 3 times to remove any surfactants on surface of the explants. Then these explants are treated with citric acid ascorbic acid solutions, antioxidant solutions for elimination or for protection of the phenolic compounds at initiation level. So generally allowed concentration level per liter 150 mg per liter. So in this solution, the circuits are treated just for 1 minute, followed by washing with the sterile distilled water twice. Then now these explants are soaking in a sodium hypochlorite solution 4%. Allowing 2 drops of twin 20 for 15 minutes, then followed by the sterile distilled water thrice. Here time allows at a different level, 8 minutes, 4 minutes, 2 minutes, totally 14 minutes. We are rinsing with sterile distilled water. After this, outer layer of the explant removing and trimming. Then next it is going to treatment with the mercury chloride for 5 minutes, followed by sterile distilled water 3 minutes, 3 times. Sorry. So the timings are also different: 6 minutes, 4 minutes, 2 minutes. This protocol standardized in this laboratory, and then after this remove, it depends on size of the explants, 1 or 2 layers of the explant material. Finally trim, and the explants just shift into surface of the media. So these are explants, trimmed explants, and allowing for M45 P20 solution. The first one shows the initiated surface sterilized explant onto the surface of the media, which allowed for or incubated for 4 weeks. The second photo shows that the response from the initiated pad within 2 weeks measures the amplification level. Then next shifting level, after 1-2 shifts at a 3 passages level, these cultures are developing the multicultures. Then inoculation and growth of the cultures. After the inoculation, the cultures are allowing in sterile zone for development under the sterile conditions. So due to the presence of the all elements, the media which is very equal to the soil, it have all kinds of elements from the macro elements to all gelling agents. This also are adjusted to pH value 5.75 to 5.80. So this shifted cultures continuously maintaining under aseptic conditions at 24 plus or minus 2 degrees temperature and 15 to 60% humidity. So this is how cultures are trimming in front of the laminar inflow by the technicians. They are separating. First, here shows the separation of the. This is the multiplication practice doing by the technicians. Separation and trimming of banana cultures in sterile zone in front of the laminar flow. Screened cultures are opened in front of the laminar flow. And then the clumps are divided into small clumps. So after trimming done by the technicians, they are shifting onto the fresh media. So here cultures are growing under controlled conditions, under light the rooted plantlets are growing, under dark, the multicultures are maintaining. Then production of the shoots, certainly the inoculated or shifted or subculture plantlets are allowed for incubation, the period of 3 to 4 weeks. So well-suited plantlets may develop. Then these well-suited plantlets are after 4 weeks are going to subculturing practice and allowing for root development. Here, auxins, generally IBA we are using for root development 1 mg per liter. In the multiplication practice BAP, we are generally using 3 mg per liter. We are not using any auxins. So here we are allowing for 4 weeks. Well-suited, well-rooted plantlets are ready to dispatch, are ready to primary hardening. So these are tissue culture, banana rooting cultures. Then hardening, primary hardening. Well and rooted. That means 5.5 centimeters to 5.60 centimeters height of the plantlets are subject to selected for primary hardening. These culture vessels are sending to grading area. In the grading area, all any debris material, agar or dried material completely removing and sending to the primary hardening for plantation of the plantlets. So here see the primary hardening level, the primary plants are growing. This photo shows completely primary hardened plants. These plants also we can sell to any customer who wants primary hardening plants. They are growing in their place and selling to native people. These are secondary, well grown primary hardening plantlets we are planting in the secondary hardening area. Maximum 32 days is required for development of a 1 feet of a plant. The entire tissue culture technology is from mother plant to complete field. This is mother plant, which is used for selection of the subcuts. Initiation stage, multiplication, rooting stage, primary, secondary, and ready to dispatch. Then what problems generally we may face? Industrial level, the problems are occurred in 2 ways only. The first one is technological level. Second level is financial level. Technological level will get the more problems, so that we'll see the more financial issues. Technological level, if you failure washing of a bottle properly, not followed, not executed protocol in a bottle washing, there will get the problem. If we failure in media preparation, there we will see the problems. And if we failure in selection of the mother plant from the different area of fields, suppose they may have some of the diseases. From the disease infected fields, if we collect the mother plants, explants, then we'll face different problems. Or we are not supposed to subculturing done frequently. The cultures are going to problems. Majority of the cultures are converting into are suffering excessive blackening. This is due to caused by the oxidation of number of polyphenolic compounds. Those are released from the wounded tissues. This may happen at initiation stage. So this may form a barrier around the tissue. It blocks the absorption of the nutrients from the media. At subculturing time also we'll see the same problem. If the technician not properly doing culturing practice, they are cutting in different way. Removal of complete entire baseline. So multiplication is the basic problem. You can't see the particular specific multiplication ratio. Generally, in any commercial laboratories, use of BAP3mg, the multiplication ratio is 2.8 to 3.2 multiplication ratio. Sometimes these initiated cultures should be kept in dark conditions to avoid release of the phenolic compounds, or addition of the charcoal. Charcoal is the best choice substitute to observe the phenolic compounds which are released by the plantlet material at the initiation stage, so that the plants definitely responds. These are the basic problem. And what are the challenges in tissue culture? This is very important role playing. There are a number of challenges before this. Technological challenges, financial challenges, technical challenges, marketing challenges. Of course, if you are sound in tissue culture technology, there are a number of ways affecting on you. The way like a technical issue. If you are not in active mode, I started, that's all. People are working, that's all. We are paying, that's all. They have to work, that's all. If you are thinking very light way, actually tissue culture technique is a very simple technique. Very simple technique. We are not using a big machinery system. Very simple equipments, instruments we are using. But here, the main issue arises. Involvement of all people who are working as a technicians or laborers at different sections. So the first challenge in plant tissue culture is the recalcitrance. This is the first challenge. Second challenge here in this, when I started this company, 2015. Now it's fifth year running. I never seen this recalcitrance problems in this particular laboratory. It's because of the media doesn't have are more rich with ammonium or calcium or salty. So cultures are inhibiting on surface of the medium. They are in reddish. Inhibitory factors are influencing on growth rate of a plantlet. So it's never happens if you are technically sound. The contamination, generally contamination may carry in different ways. Through the sterilization practices, whatever utensils we are using in sterile zone, you must be sterilized before. Our movement of the persons who are working inside the sterile zone, they must be clean. Dress wear. The contaminants generally will see the inside the contaminants, bacterial, fungal contaminants, airborne microorganisms, sometimes endophytic bacterial species also causing damages of our stock cultures. This will be happen in selection of the mother plants. Phenolic browning, this also, and some of the seasonal variations, suppose summer, winter, then rainy season, the temperature fluctuations in temperature, humidity, moisture content inside the laboratory, outside the laboratory, seasonally variations. And the dehydration of these plantlets also called vitrification inside the laboratory, and some of the somoclonal variations. See a few points here mentioned in recalcitrance. Generally, due to loss of morphogenetic competence and totipotency capacity. Everybody knows about the totipotency. A single cell, it can be produced an entire organism through the differentiation process. It is the metabolic pathway which is involved to convert a single multicellular cell into a complete multicellular organism. It may be animal or a plant. The loss of morphogenetic competence means changes in genetic material, proteins, enzymes, etc. These macromolecules are affecting, influencing by the some other agents. So that we'll see the reducing the totipotency characteristic inside the lab, inside the culture. This will be happen due to the free radicals are occupied with the paid electrons. So that damage of the complete plantlet material. So the major solution is that very younger tissues or juvenile tissues you may subject to select before going to selection as a explant of a material. This is a recalcitrance. I never seen in my laboratory this problem or this challenge. The contamination is I faced the contamination problems at second and third year. The first contamination due to maintenance of my laminar flows inside the laboratory. I got phenolic dark cultures inside the growth room. The second contamination also faced in second year through the washing section, not properly washing the bottles. The third problem faced in my industry because of the problem of autoclave, fully automatic autoclave, fully automated, temperature maintenance, pressure maintenance, automatic, fully automated. These three problems given more contamination in second and third year of my company. Because of I trained people, they are doing okay, just I am checking and going. So earlier I mentioned that we should be in an active position. So this maybe by following different, wear gloves, lab coat, whitening of our hands with 70% alcohol, working benches, and the area. The next one is generally phenolic browning. Many secondary metabolites or phenolic compounds are releasing. This not happened in multicultures. Generally, at the time of initiation, this may be happened. But hardwood species are very difficult to grow inside the laboratory. These may are releasing the secondary metabolites. Everybody knows about the secondary metabolites which are present in medicinally valuable plants. So this may releasing phenolic compounds. So this those are inhibitory agents to grow of cultures. Seasonal variations because of the humidity and the dry season. The nutrients in summer generally nutrients may go into evaporation. The extreme moisture level, this media is going to occupy with some of the surface contaminants like bacteria, some species. Sometimes direct air enter into the environment, enter into the sterile zone. This may affect the sterile zone maintenance practices. Sometimes some may insects enter into this seasonal vitrification. This this is caused by the hydration, excessive hydration, and low lignification, and it reduces the mechanical strength of the tissue culture generated plantlet. And some of the oxidative stresses like high salt concentration, high relative humidity, low light intensity, more gas accumulating, not exchanging of air between bottle, inside the bottle and the outside the bottle, means bottle and the growth room zone. And the amount of low calcium content inside the media supplemented with generally calcium chloride we may prefer as a calcium element. And the length of time we are allowing for subculturing practice. Generally, we should take care of this subculturing practice between 4 to 6 weeks. And sometimes higher amount of ammonium concentrations also contributes to hyperhydricity. And the somoclonal variations, this will be happen due to some of the addition or deletion of the chromosomes in genetic material, and it changes the phenotypic characteristics. They are not stable, heritable. So here we need to avoid long-term cultures. Here we have to take up, or we have to see that multiplication cycles. And avoid addition of the any 2,4-D. Generally 2,4-D using for induction of the callus. Undifferentiated mass of callus may generate addition of the 2,4-D to the media from the organized tissues. What is the Swot analysis? What is the strength of banana tissue culture? What is the weakness, opportunities, and threats? Strength is generally we have to ask question ourselves. I have capable to carry out this technology. I have capable to execute this technology. I have capable to contact. I have capable to convenience any customer. I have capability. Any different small innovative technology adapted in mind, because it is a cost effective product. It's not a luxury industry. It's a laborious work. Number of people are working at different sections. So the strength is technically we are sound. If we are sound technically, then next step is command on market. So here customers are farmers, not any other persons. There are in between producer and in between customer. There are number of agents, dealers, distributors are occupied. And financially, are we strong? This is also one strength. And we should know the climatic conditions at agricultural practices at different areas, national, international level, local level. For cultivation of any kind of tissue culture plant, tissue culture plants. And also in terms of quality, are we producing very uniform plantlets? Are giving high yield like this? A number of financing institutions also supporting and also interesting to give financial support for commercially producing banana plants. These are climatic conditions and the type of plants we are producing, and then financial level. This strength, weaknesses. The first weakness is initial level is the investment. It requires more investment. Very simple technology, but investment, more investment. The technology also relatively sophisticated. And the need here, skilled person, knowledge persons with different areas. What are the opportunities? Due to demand of banana, the favorable conditions like state level government policies, central level government policies, national level government policies, like our international level government policies. That is the first opportunity. A demand, day to day increasing the demand, only not reducing the demand, particularly to the banana. Marketing potential, have to occupy in all areas, national level, international level. These are the opportunities to us to start a small scale level industry. And there are number of threats, competitors, number of competitors. We have to show our competence to the competitor by encouraging them. And what are the environmental factors influencing on laboratory level, hardening levels, acclimatization level, and field level? And sometimes fluctuations in the market, local market. See, in this time, corona issue, major fluctuations in banana markets. Farmers are suffered a lot. They sold out 10 rupees per bunch. Sometimes price also. These are threats. The capabilities required. Suppose I myself turn to as an entrepreneur. Knowledge, initiative, risk-taking ability, planning of business, hope of success. Communication skills means to the customer, farmer communication skills here. And then next is what are the significant challenges, market-related risk? Generally, it happens. If I start a company here, generally, I can see the market-related risk. When we going to specific area, they may ask where you given plants, show your plants, what is the yield, how they are producing, what is the bunch height, what is the fruit size, is it suitable for export or local? That's definitely happened, market related issue. List. Locally, state level, different state level, intra-state level. Then access to finance and the credit. It depends on sale of plants. Definitely it involves access to information, low bargaining power, then economic sometimes shocks, and access to training and related challenges. Then I want to start a tissue culture commercial lab. What are the main requirements? Infrastructure level. Tissue culture laboratory infrastructure is required. Here are different sections which are functioning. Washing section, media section, inoculation section, growth room section. In washing section, we don't want require any equipment except only RO water. In media section, we need balances, simple balances, electrical balances, and a few small equipments like pH meter, cyclometer, magnifier, homogenizer, then RO system, autoclave. In laminar flow, in inoculation room, generally few benches are, laminar flow benches are required. AC maintenance of the temperature and the maintenance of the control conditions inside the sterile zone. ACs are required. For certification of the laboratory purpose, maintenance of the 1 lakh lux sterility conditions inside the laboratory, pressure modules are required. And bottles, culture bottles are required. To hold the bottles, trays are required. To close the bottles, caps are required. So growth racks, etc. And to provide hardening facility, there generally shade net is required. One frame structure is required. And the PP sheets, poly bags, and generally consumables. These are the infrastructure we required for establishment of a tissue culture laboratory and equipments. The chemicals, like generally in media section, different types of chemicals are using for preparation of the media, which is almost equal to the soil, having all kinds of macro, micro, growth, vitamins, everything at a different concentration. And also nursery level, also some of the chemicals are required for growth of the plants and control of the insects and the pests. And the manpower, these four are required to start any tissue culture facility. And what's the estimated project cost for a TC mode, TC tissue culture model? Suppose you want to produce 1 million capacity of a plant tissue culture size. The laboratory size is required 5,000 square feet. Sterile zone, non-sterile zone, all sections which are occupied in this 5,000 square feet area. Greenhouse area almost 3,000 square feet. Here the sections like primary hardening, secondary hardening, dispatch sections. This is the fixed cost for complete unit size. The fixed cost for tissue culture laboratory is 40 lakhs only for tissue culture laboratory purpose. And the laboratory purpose, equipment purposes, 35 lakhs. The other requirements like office, furniture, fixtures, 2 lakhs. Water supply system in both tissue culture facility and the greenhouse facility, 20,000, and greenhouse and shade house equipment purposes 10 lakhs. Generally, protocol is available any commercially available. We can purchase it as per demand, 2 lakhs or 3 lakhs, depends on recurring cost. Generally, all consumables, maintenance charges, everything all are 7 lakhs. It's only for a year. Total 26.2 lakhs. Saleable price cost is 10 rupees. As an entrepreneur, if we convert into as an entrepreneur, we are producing, that's why we are an entrepreneur. We produced a product. So we have to show our quality in terms of our services, our leadership, our capabilities. We have to face all types of the risks, responsibilities, both losses and profits. Always should be passionate about growing our business. Every entrepreneur is always innovator, thinking continuously, not going to stop. Small change. Every entrepreneur looking for a better future, putting more efforts and thinking always profitable ways. Worldwide, the entrepreneur is going to associate with the economy, major political environment, social environment, weather. The next circle by the entrepreneur, technologically support, financial support, services at commercial level, market, agents, extension workers, who are the competitors, and government policies. The government schemes and incentives who are going to start up a tissue culture unit in India, APEDA, Agriculture, Processed Food Product Export Development Authority, NHB, National Horticulture Board, DBT, Department of Biotechnology, SFAC, Small Farmers Agribusiness Consortium, and the state level incentives. They are giving different subsidy levels by the national level, at a state level, and by formation of consortium. So here I am going to concluding my session. So the technology has been very successful as an industry to produce any kind of plant species, not only banana. It is continuously contributing successful agricultural practices, not only for commercially producing, to preserve any endangering species through the indirect organogenesis. As we discuss now, complete it's a direct organogenesis practices applying in tissue culture technology through the micro propagation method. And a number of Indian farmers also becoming aware about the propagation of not only banana, the stick plants, then date farms, then flowering plants, then ornamental plants. So, new entrepreneurs having a good scope in production of the banana tissue culture through the tissue culture technology by applying micropropagation technology. So with this, I conclude my session. Thank you, sir. Having any doubts or clarification?
Sir, can you stop the screen sharing?
One second. Okay.
Yeah, close it again.
Hello.
Thank you so much, sir, for keeping our session from 2 to 3:40, and all our participants, I hope they were from beginning to the end, they were listening your lecture, and there are few questions. The first one is, how would the contamination be occur in the laboratory? How to overcome the contamination in the laboratory?
Through the fumigation techniques, sir. Through the fumigation practices.
Fumigation.
Yeah.
Fumigation with aldehyde or?
Fumigation with potassium permanganate and formalin solution. 2 grams of potassium permanganate, 10 ml of formalin solution. So we can eliminate the contaminants through the fumigation.
And
mixing with formalin solution. Formaldehyde.
Do we to do periodically, or do we need to practice this one periodically for a certain?
Weekly once, sir.
Okay.
Weekly once.
Right. But formaldehyde generally so weekly once means it will be more hazardous in terms of like
only Saturday we are practicing. Only Saturday we are practicing, so not in between working days. Only weekend days. The Sunday complete closed.
All right, weekend you are practicing.
Weekend.
Yes, sir. Yes, sir. Estimate cost of equipments required. So there was a question, estimate cost of equipments required for this banana tissue culture.
Only particular banana plant tissue culture.
Yeah.
Estimation cost almost 35 lakhs it will come, sir. Including all growth racks, 35 lakhs. Growth racks, pressure modules, air conditioners, autoclave and all bottles.
So is there training opportunity? How one can apply the training opportunity in your laboratory? Is there any opportunity, they were asking?
Okay. Generally, we are not allowing any opportunities here locally available. For training, we are allowing for 1 month period, sir. 1 month period. Generally, charges we are taking 8,500 rupees.
8,500 rupees for 1 month.
For 1 month.
And their eligibility, sir, is there any eligibility requirement?
Any science graduate. Any science graduate.
Okay.
Any science who are interested.
Okay. Some more questions, sir. Is there any internship for BSc agriculture students? How we can apply for it?
Internship. Generally,
the last
directly
question.
Internship or incentives? Internship.
They are asking internship.
For AGBSc students, agricultural students.
Yeah.
Directly you can apply, sir. No problem.
Okay. Directly.
How much period, sir?
Yeah. So.
Period of time?
No, they were asking. I do not know. I think Sani Komu. Sani Komu, you can reply for that.
Ranging from 1 month to 6 months.
Is it 1 month or
8th
2
sum of
months?
agriculture. Maybe 6 months or 3 months.
Sir, for internship program, we are not charging anything. For internship program,
Okay. All right.
particularly for training program, who are asking
Hmm.
to get to train in tissue culture technique only we are charging. For internship program, we are not charging.
Sir, if we get any tissue culture banana plants, will you send to us?
Yes, sir.
Okay. So I think you can provide your email or your contact details so that I can share with other people. They can.
Sure, sir. Sure. Already given this my visiting card to.
Yeah, many of them they were asking for training. Training information like how
I
can
am
we apply,
selecting
how
few
to
people
contact
only,
it.
sir. Sir, 1 second. I am selecting only few people who are
Okay.
interested tissue culture, not all. Who are particularly
Okay.
interested in plant tissue culture practices only, I am encouraging, not all. For internship
Okay.
program.
Okay.
That's why I am not going to charge.
Okay, dear participant, I think I'll unmute all of you. I'll give the privilege if you have any queries and if you want to interact with the facilitators, so you can just unmute yourself. You can talk. Okay. Yeah, now you can unmute yourself and you can talk. Hello. Sir, you can unmute since I have given, so
Yeah.
it's
Okay,
automatically muted. Any participant you would like to ask any question?
Good evening, sir.
Yeah. Yeah, tell your
Sir,
name.
how can we apply? My name is Sharif and I am from Sanskrit University, Mathura, Uttar Pradesh.
Okay. Okay.
I am studying BSc Agriculture 3rd year now.
Okay.
If we want to go for internship regarding tissue culture, how can we apply, sir, in 8th semester?
Okay, I will provide an email. You directly apply to my mail ID. Then I will confirm you later. So,
okay, sir. Thank you, sir.
Get a letter from your head or your dean. Send it to me. I will respond you.
Okay, sir. Thank you, sir.
Yeah, any other participant? If you have any question.
No, sir.
Sir, there is another question, sir.
Yeah.
Is this tissue culture can be applied in vegetables also, sir? And why we are not using tissue culture much in vegetables, sir?
It's not commercially viable. Cost of production. We check it out. Cost of production. If it is commercially viable, then we'll go. We'll do. No problem. Which is the commercially feasible, viable? We have to select those plant species from. All species technology is available, but which is the commercially valuable and the market, local, surrounding, national,
Sir, there was a question. What has been the impact of government subsidy in tissue culture in India?
Impact?
What has been the impact of government subsidy in tissue culture in India?
It's a specific to tissue culture laboratories or tissue culture growing formats. There are two government, central government giving subsidies to tissue culture rising persons and tissue culture units. That's specific to only industry or farmer who are grower.
I think in there the question goes like a laboratory aspects. I think so.
Aspects.
Or in the cell.
Okay. NHB.
Okay, so I'll ask thing. Robert, can you clarify it whether it is from the laboratory side or this
industry side
selling
or
point
consumer
of
side?
Industry side, yeah.
Industry
Robert,
side.
can you call? Robert, can you speak? No, I think you can answer both the sides, sir.
Okay, industry side, NHP giving 50% loan subsidy. Suppose the tissue culture facility we are developing up to 3 crores. The government of India through the National Horticulture Board mission, they are giving 1 crore 10 lakhs directly subsidy giving to the industrialist. At the same time, to the farmers also who are growing tissue culture plantlets, not only banana, some other species, they are NHP providing subsidies. They have to apply through the proper channel.
Okay. Sir, one more question: whether tissue culture is possible in gerbera flower?
Already, in number of gerbera plants are producing through the tissue culture technology. A few labs are producing. Protocol is available. Standardized protocol is available.
Okay. Yeah. Madam has given the email. Somebody is asking Madam Saraswati email, and she has flashed it in the chat. I think you can note of it. There's somebody asking. Sir, you can type your email also in the chat box so that others they can.
1 second.
They can have a contact. Just you can type it in the chat box. Yeah, Madam has given the number also. You please note it down. 63801413. Yeah, please note it down. It is there in the chat box. I think you can copy and paste. Yeah, I think before, if you have any questions, please let me know. Otherwise, we are going to conclude. Before concluding that, before proposing the vote of thanks, I would like to give some announcement to the participant. We will give the feedback form along with the questions to you by the organizing secretary, Dr. Prasant Nimbulka. He will be sharing all the details to you along with the YouTube link as well as this contact details of this resource person. And if you have any further doubt and clarification, you can even directly contact with them. And so you will get to the certificate within three to 4 working days, because we need to process it. And so a lot of things are there. So be patient. Those who are attended last webinar also keep on asking when we will get to the certificate. We understand your need, but we have some with time to process. We need some more time. So please bear with us, and within 3 to 4 days, we will send the all the certificate to each and every one. Okay. So thank you so much, sir, for your elaborative technical details about this banana tissue culture. I think participants, they have benefited. Sir, do you have any videos, sir, in your YouTube regarding your companies and your laboratories? Sir, can you unmute? It has been muted because some background voice I have muted on.
We don't have any video. You just go through website, sir. miccolabs.in. MICCO.
Okay. In the website it is there.
Website is there. miccolabs.in.
Okay. Okay.
miccolabs.in.
Right. Okay. Sure, sir. We'll see that. So thank you for your information and taking pain in elaborating all this to our participant. Thank you so much, sir. Now I'll request Dr. Tasayatan to propose the vote of thanks. She's a coordinator of this one day's webinar.
Yeah, thank you, sir, for giving this opportunity to propose the vote of thanks. Good evening, one and all. Okay. So I would like to thank Dr. Basanta Singh, sir, Deputy Director of Instruction, Central Agricultural University, Imphal, for his constant support. Professor B.N. Hazarika, sir, Nodal Officer, and Dean College of Horticulture and Forestry, Pasinga, for being our guardian and always guiding us and for being our guiding force. I also would like to thank Dr. P. Raja, Associate Nodal Officer, IGP NAHED CHF Pasinga, for his continuous encouragement and helping us in conducting the webinar. I owe my sincere thanks to our eminent speakers, Dr. M. Saraswati Ma'am, Principal Scientist, ICR NRC Banana Tiruchipalli, and Mr. Dharneshwara Reddy, Managing and Technical Director, Miko Laboratories Private Limited, Andhra Pradesh, for accepting our request and sparing the time from their busy schedule to share their knowledge. Not to forget our participants who have been with us throughout the day. I thank you all. I thank all the committee members who have been working day and night since weeks to make this program a success. Last but not the least, I would like to thank all the staffs of IDP NAHED projects HF Pasinga who were giving their support in all possible manners to make the program a success. Thank you once again for staying with us and making this program a great success. Thank you.
Thank you, Taso, for proposing the vote of thanks. Finally, I thank you, Saraswati Madam and Reddy Garu, for sparing your time and enriching our participants. Thank you so much. With this, and before ending, I would like to take a photo snapshot. I request all the participants, can you start your video? We want to see your faces, and a resource person also, they want to see your face, okay? So please start your video. Yeah, okay. Start your video. Okay, thank you so much. Thank you, one and all. I'll end the meeting now. Thank you.
వాసుకుతాను